MRI analyses are kind of like this. What I hope to do with this class is take imaging analyses from this to this. No, seriously, what I hope to accomplish with this class is give you a foundation upon which you can build more skills. And so I've spent the past 12, 13 years learning how to do imaging analyses. None of that was in a class. What I hope to do with this class is to share a little bit of what I've learned and kind of give you an appreciation, at least, for the complexities of imaging analyses. So then when you read research articles, you can appreciate a little bit more what goes into the research. And then this class can also serve as a foundation for those of you who might want to go on to do imaging analyses as part of a research career. To start, we're going to first give a brief overview of neuroanatomy. This is the neuro part of neuroimaging and really why we care about the MRI analyses. This allows us to know where we're looking in the brain. So our first introduction is understanding our different planes and directions in the brain. We have directions: superior or dorsal, inferior or ventral, posterior or caudal, anterior or rostral. And those are our primary directions. They're shifted in the brainstem. In the brainstem, dorsal is toward the back. So we think a dorsal fin of a dolphin or a shark, that's on the back and so dorsal to the top for the human brain, but brainstem, spinal cord toward the back. And in that case, I really prefer the posterior and anterior. It's just a little clearer. And then we also have three main cross sections of the brain. And these are important with imaging because they are how we acquire scans. And they would be either in the coronal, which is our frontal section, is another name for it, so like a crown. We also have a medial section in this case, but the type of section is called a sagittal slice. And then we also have horizontal or axial or transaxial. There are a number of names for each of these, but those planes are important to understand just so you know what you're looking at, the brain. And another view of them. Again, coronal, we can remember it's like slicing across, back to front, front to back, but like you would have a crown. Horizontal is parallel to the ground if you're standing up, and then sagittal. Just different ways to remember them. And we can see here there are examples of the different types of planes. So we have sagittal, coronal, sagittal, and then here would be an axial. And so again, it just helps you be oriented. Now we're going to do a quick overview of the brain. So this is the brain from the top. Eyes would be this way, pointing toward the top of the screen. And cerebellum would be in the back, down here toward the bottom. Now this is a false color image of the brain, obviously, but this was created using one of the tools we will use in this class that allows us to take an MRI and process it and be able to look at a number of different regions, either volume or thickness or some other metrics. But there's structure to it. So those parcellations, as they're called, are really structural groupings and areas of the brain, but there is really a lot of function to the brain. It's not just, we need to understand where something is, but also, importantly, what it's doing. And we have certain areas that are what we call primary sensory cortex. And so we have, here's our primary somatosensory, primary visual, auditory, olfactory would be on the temporal lobe, we have primary motor. So these are just very basic motor sensory cortex. And then as we spread out from those, we start to get into what we call unimodal association cortex. And these are areas of the brain that are, for the most part, there's not strict rules with all of this, but for the most part, these are just doing one thing. So just behind our primary somatosensory cortex, we have this somatosensory association cortex, which means for the most part, this part of the brain is processing this sensory information. And then same thing, motor, just in front of motor cortex, we have this primary, this area of the cortex is primarily involved in just motor functions. It is more complex than that. And then as we spread out from there, we get into heteromodal association cortex. So the cortex is quite large, but compact in structure and in storage. Our brains are only so large, and but if you took the whole surface area, laid out the cortex, it would be about 2,200 square centimeters, or 2.5 square feet. And most of the cortex, the surface, is hidden within the sulci. So those are the grooves. We have the gyri are going to be the bumps, sulci are the canyons, the grooves. And so about 2/3 of the cortical surface is down within the sulci. So again, if we're just looking at the outside of the brain, there's a lot that we're missing. And that's where the imaging comes in, because then the imaging can look all the way down into the sulci. And the thickness of the cortex ranges from about 1.5 to 4.5, generally about 2 to 4 millimeters. It's thickest over what we call the crest of the gyri, that's the top part of the gyrus. And it'd be thinnest at the bottom, but different parts of the brain are thicker and thinner. We tend to have thicker cortex in primary motor areas and some of the primary somatosensory cortex. And then other areas of the brain are going to be a little thinner, and that's just sort of natural. The overall weight of the cortex on average is about 600 grams, or I shouldn't say mass, and it's only about 40% of the total brain weight. And then of that, only about a third, a little less than a third, are going to be the neurons themselves. The rest are going to be glial cells that are providing a lot of support and other functions. And their specific functions are outside the purview of this class. And one other thing for background that's important to understand, this is more important if you do any developmental research or aging research. Because what happens is we have certain areas of the brain that are that develop earliest, and those are going to be our primary motor and sensory cortex. And then as we spread out from there, we quickly get into areas that tend to develop a little bit later. And but in particular, the frontal lobe is going to develop the latest. But as we age, that is also the area of the brain that tends to atrophy and change with age earliest. So it's this last in, first out principle. And so we have these certain areas of the brain that tend to be more affected by aging, just what we call normal aging, and also the latest to develop in childhood and adolescence and into adulthood even. But that can be important to understand, because if we're looking at normal or abnormal development or normal or abnormal aging, these are areas that we should be aware of this pattern. And the areas highlighted in 1, our primary motor and sensory cortex, tend to be relatively unaffected by age. So if there are changes in those areas, it could be due to some neurodegenerative process or some other damage, although that's not always the case. That's the cortex. We have a lot of white matter. So that's just the outside of the brain. So inside we have connections between areas. And there's a number of large pathways. We'll get some experience with some of the pathways with our diffusion imaging analyses that we'll do later in the semester. But the pathways really are not as simple as that previous image showed, very complex. So this was created using some tools that we really won't get a lot of experience with in this class, some more advanced imaging. But this is diffusion imaging and showing even just a handful of the different connections. We can't image the axons, they're much too small. So really we're imaging bundles of axon and big bundles of fibers. And even this has all the complexity of this image, there's still a lot more going on in the brain than this will show. But the colors are just denoting direction. Green primarily means front to back movement, anterior to posterior, blue, superior, inferior, and red is left and right, or in and out of the screen in this case. So we have gray matter structure that we can analyze, and these were created using tools we'll use this semester. We have white matter structure we can analyze, and we can look at strength of connections between regions of interest. So part of the frontal lobe over here and the thalamus, or we can look at overall networks in the brain. We can put everything together to look at gray and white matter. So this is just an image from one of the research participants, but looking at connections between frontal cortex, the frontal lobe, and the caudate nuclei, which are some deep gray matter structures important for cognition and motor function. We can put everything together, and again, we can look at gray matter, we can look at white matter. So that's generally my approach for a lot of projects, is not only look at just the gray matter, but also look at white matter, because all of the connections between the areas are going to be important. You can also do, we can look at networks functionally. So this is an analysis looking at resting state functional MRI. I'll talk about that a little bit more. But we have what we were showing previously, all the structural components of the brain. Gray matter, thickness and volume, white matter. We can look at the pathways, the white matter connections. And here we can also look at connections between different parts of the brain, but using this functional imaging. So this is looking at patterns of blood flow and how are those patterns of blood flow correlated throughout the brain. And can use that to set up or to really understand a lot of different networks we call functional networks. Now we're going to transition into a little bit more of the anatomy. I know this is sort of a whirlwind. You can go back recording and slow it down or speed it up, kind of ignore it altogether, whatever your preference is. But there are other parts of the brain that are important to understand. One of these is the meninges. These can affect our structural imaging, which is why I talk about them in this class. There's three main components to these. This is the covering, or the sac, if you will, this bag around the brain, and composed of dura mater, the arachnoid layer, and pia mater. And the dura mater is the outermost layer. It's tougher, it's very, can be very thick, difficult to get through, so it provides good protection to the brain and holds everything in place. But these could show up on our imaging. So there's a picture of the dura mater, and again, when we do what we call segmentation of the brain, it'll be done automatically by some software. But there are areas where this dura mater is really close to the cortex that it will get picked up, and the software will say, oh, this is cortex, this is brain, when it's not the case. Then there's the arachnoid layer, which we won't worry so much about in this class, but this is the middle layer of the meninges and is very important for processing the flow of cerebral spinal fluid. So it's important with the clearing of metabolic byproducts and waste. This is some of the role that cerebral spinal fluid is doing. Then there's the pia mater, the reverent mother, soft mother, that's right next to the cortical surface. It follows the gyri and sulci, goes down everywhere and is adhering very closely to the cortical surface and really is indistinguishable from the cortex. So where are the meninges on this image? If we're looking here, this is our mid-sagittal slice right down the middle of the brain, and so right here we're seeing a little bit of dura mater. So this is what we call skull-stripped brain, meaning we've removed the skull, and so now we can just look at the brain. And right here, let me draw a little bit. There's some dura mater. This is meninges, and right there. So we can see some different meninges on here. Now, for the most part, it's outside the gyri, and so you can look at it and you can tell. One of the questions is, does the software, the imaging software, can the computer tell what this is? And generally not. Sometimes it can. It's getting better. But there again, there's meninges down here, right around the brainstem. And there is also meninges right here next to what we call the medial temporal lobe. And that becomes a potential issue with some imaging. So that's something we're going to learn to deal with. Again, where's the meninges? This is zooming in. So this structure here highlighted in red is what we call the entorhinal cortex. This is primary input and output to the hippocampus. So this structure in red is very important for memory. It's important for some other functions as well, but memory is one of our important ones. So if you have software, we want to measure the thickness. That would just be how many millimeters thick is it from outside surface to the inside. What can be important to understand, as I drew on the previous slide, is there is meninges right there by it. So we have cortex right there, but just outside of it we have meninges. And the software that we use and that we'll learn to use that will automatically pull out this region and kind of label these different regions of the brain tends to have some issues with that. And it usually tends to have that problem in labeling correctly, distinguishing between cortex and meninges, the dura mater, sort of across the board for everybody. So it's okay. It's essentially reproducible error or reliable error, but it is still error, and it can make that part of the brain look thicker or plumper, larger than it really is. So that, again, bring up meninges because it can cause some issues with our imaging, but for the most part, relatively minor. So if we're dealing with large data sets, and a lot of people generally don't worry too much about it. Another part of the brain is the ventricular system, and this is really important for imaging. We have these ventricles. We have 4 ventricles, 2 lateral ventricles, a 3rd ventricle, and then the 4th ventricle down by the cerebellum. And within these ventricles, we produce cerebral spinal fluid, and that is an important product, mostly water, but it helps cushion the brain, it surrounds everywhere in the brain, it helps extract the metabolic byproducts, waste products, potential toxins, all of that. And the ventricles, though, can be important because they are at least an indirect marker of brain size. And what I mean by that, particularly with aging, is as we get older, our brains tend to shrink somewhat, and with normal aging, we're not losing neurons, we're just losing connections between neurons, and we can lose some myelin. So we have some loss within the white matter. A lot of things are going on, but to compensate, the ventricles get larger. So ventricles at their core are going to be, if you're looking at an abnormal brain or a normal brain, they can tell us a little bit about what shape the brain might be in overall. And so there's something to pay attention to. We'll skip the video. But here's an example. So over here on the left would be what we call normal lateral ventricles. And normal for someone who's a little older. And if we look at, if you're 20 years old or up to about 40 or so, the ventricles for the most part are going to be quite small. In some cases, barely noticeable. I mean, they're there, but depending on the scan, not very large. But as we get older, so getting into our 60s and 70s, they start to enlarge. But this is kind of normal for aging. Over on the right would be abnormal. And this is due to what we call hydrocephalus. So this water retention, the cerebral spinal fluid in the brain, and there's other issues going on, but this very large ventricles. So we see ventricles that large, generally we think hydrocephalus. It's possible someone could have a lot of atrophy, have lost a lot of brain matter, and have very large ventricles, but generally would not be very functional. And if we look around, this person doesn't have a lot of atrophy. There's not a lot of space around the gyri, the sulci are not particularly enlarged. So in this case, this is a hydrocephalus. So what can be done? Neurosurgery, put in a shunt, go and drain out the cerebral spinal fluid, ventricles get smaller, and function tends to return to normal or mostly normal. This person has some other issues as well. This darker area right here indicates some damage. It could be reversible, but that is certainly some damage there. Then another part of the brain that we could be concerned about or interested in, depending on research questions, is the limbic system. This is these core, generally kind of evolutionarily old areas of the brain, and it would include the cingulate gyrus, the hippocampus, parahippocampal gyrus, the amygdala, and these are all areas of the brain that are involved in emotion and memory. And just as I was talking earlier about white matter, we can look at different gray matter parts. We can look at the hippocampus, but there's a lot of white matter pathways. There's this part that comes off. This is white matter called the fornix, and that comes off the back of the hippocampus and wraps around, travels on top of the thalamus, and goes down to mammillary bodies. It would be part of what we call the hypothalamus. These are involved in a lot of basic functions, including regulation of hormones. And then there's other connections up to the thalamus. So these are white matter pathways. And so you might have a research question. You're interested in memory or emotion. And so you could look at some of these pathways. And there are ways, some of the tools that we'll be using, that allow you to look at the integrity of these systems, to look at, again, essentially what shape are they in, how strongly do they connect between areas. And So these are white matter pathways within the limbic system. Another part of the brain that might be interested in studying, and we'll get some experience with getting volumes out in this class, or be the basal ganglia. And these are groups of gray matter structures within the brain, kind of deep within, buried in the white matter, right in the center of the brain. Basal ganglia are made up of different structures. The three structures of the basal ganglia would be the caudate nucleus, putamen, and the globus pallidus. And these can be segmented from the MRI. They can be done manually, or they can be done using different imaging tools. And it's sort of an interesting process to do it manually. I did some of that in my first years as a graduate student. Sit there and have to trace, draw. We have the brain MRI, and sit there and use software to trace these different structures in the brain. And it's a good way to learn the brain, but it takes a long time, and especially if you have to do all of these different structures and do it for hundreds or thousands of MRI scans. It's going to take too long. No one wants to do that. And so we have software to do it, and software is very good now and can do it very quickly. And so this was just a comparison of some different software tools. And again, FSL and FreeSurfer are two that we will get some experience with. Then right by the basal ganglia, right in the middle of the brain, would be the thalamus. We might talk about the thalamus as an entire structure. We can look at the whole volume of it, but it has a lot of different nuclei. There are different components, different parts that are doing different things. And so this is a drawing, just a schematic of different areas of the thalamus. And there are imaging software, there are tools we can use. We won't in this class, but there are tools we can use that can pull out and sort of predict where these different subcomponents of the thalamus are. And that can be important for research as we get better imaging, better software, better tools, moving into machine learning and artificial intelligence, which we don't do in this class. Technically, some of the tools we use are machine learning, but that's not the focus of this class. But as we advance, as our understanding or technology, we can get better understanding of the brain and more extraction of more data from MRI. And so now we have software that can pull out different areas of the thalamus. And again, if you had interest in the visual system, then maybe you would look at the lateral geniculate body. Or if you had interest in sensation, pain, or other feeling, or how we move, then we could look at these lateral portions of the thalamus. Or if you're interested more in cognition or some planning or memory aspects, then we can look at other areas of the thalamus. So again, these are all of the understanding. A little bit of the anatomy helps us care about the MRI. We have an MRI. Really, why do we care? It's so we can look at the brain, so we can look at behavior. In closing, I gave a very quick overview of the brain, of neuroanatomy. Can go much more in depth. This was just a very fast overview to give us a little bit of the background of where we're going to be talking about. We're focusing on the brain, so it's good to understand at least cursory parts of the brain, and but really to understand as we use the imaging tools and learn about these imaging tools. Really, we're learning about them is so you or someone else could understand the structure of the brain, the function of the brain, the behavior of the brain. And so this was our overview just to give you a background and how you can look at the brain, what questions you can ask, depend in part on what data you have available. And if you're designing a study, it's important to understand, okay, what behavior am I interested in? What parts of the brain or functions of the brain am I interested in? So what data do I need to acquire? And what tools are available to process those data and to be able to run my statistical analyses. And so that's what this class is about, is providing you exposure to some of the tools and some of the analysis methods so you can go on and do your own research, or at least appreciate the research and the work that goes into imaging studies.